nucleic acid structure and organization. This is the beginning of your biochemistry and molecular biology book. Now, this slide shows the central dogma of molecular biology. We've revised it over the years, but we used to believe as scientists in the 1950s, 1960s, 1970s also, we used to believe that DNA coded for RNA and RNA coded for protein. And we believed that that was the way that genes were expressed. That's how you make protein, and that's true. That is actually true. But there's some exceptions to that rule. Okay, and we're going to talk about those tonight. Now, you need to know, students, that the word for using DNA to code for RNA is called transcription. And the word we use to describe RNA coding for protein is called translation. Don't get them mixed up. They both start with a TR, and I get those mixed up once in a while. You can't do it on the exam. Because on the exam, they might ask you, what is the transcription product of this process, or what is the translation product of this process? So you need to know what those words mean. Okay, now if you look at this diagram, we all know what DNA is, and we're going to talk about that tonight at length. And we all know what RNA is. We're going to talk about that at length tonight. And protein, we're going to talk about that in the next couple evenings. Now, DNA, I used to teach undergraduate students at a college, and students would always come up to me and say, DNA is converted to RNA. RNA is converted to protein. And that is not true, students. DNA is not converted to RNA. RNA is not converted to protein. As we're going to see later tonight, DNA codes for RNA. The DNA is not consumed in that process. After you use DNA to code for RNA, you still have the DNA left over. Okay? And when you use RNA to code for protein, the RNA is not consumed. You can use the RNA again to code for more protein. Okay? So the process of, it's like when you take a CD, a compact disc, and put it in a disc player and play, it codes for music. But you can take the CD out and hold it in your hand, and it's not consumed. Okay, you can reuse it, reuse it, and reuse it. Such is the case for DNA and RNA, and we're going to see that later tonight. Okay, so DNA coding for RNA is transcription. RNA coding for protein is translation. Now I'm going to tell you how to remember that. I'm going to tell you how I remember that. When you go from DNA to RNA, that's all nucleic acid. When you go, okay, and that process is called transcription. When you go from RNA to protein, RNA is made of nucleic acids, but what are proteins made of? Answer that question in the classroom discussion. What are proteins made of? Amino acids. It's very basic. But when you go from RNA to protein, you are going from the language of the language of nucleic acids to the language of amino acids. So you're essentially translating from the language of nucleic acids to the language of amino acids. And that's how you can remember that it's called translation. Okay, but you need to know the words transcription and translation. So the central dogma of molecular biology is when you use DNA to code for RNA, and RNA ultimately codes for protein. That process collectively is referred to as gene expression. So gene expression is a term that refers to transcription and translation, or essentially using DNA to ultimately code for protein. You need to know what gene expression, the vocabulary word, refers to. Okay, now, as we're going to see later in this chapter, DNA can also replicate itself. This is important when you have two cells that divide. If you have a cell that decides it's going to divide into two daughter cells, you have one cell with 46 chromosomes that divides into two daughter cells, each of which have 46 chromosomes. So how do you double the DNA? DNA can replicate itself, and we're going to talk about that tonight. That's commonly tested on the exam. Now, I know what you're saying right now. You're saying, gee, this is really basic molecular biology, and it is. But they ask questions about it on the exam. And we need to start here to make sure everybody's on the same page and has a concrete understanding of the fundamentals of molecular biology before we go on to the next topic, which is medical genetics. And again, I know this stuff is basic, but it is tested on the exam, and I want you to understand the concepts of molecular biology and get points right on the exam. Okay, now, one of the exceptions to the central dogma of molecular biology, and I remember in my lifetime when this came about, it was about 1980 when we elucidated the structure of the HIV virus. Prior to elucidating the structure of the HIV virus, we did not know that reverse transcription took place. But there is an enzyme in nature in the HIV virus, and actually there's a similar enzyme in humans, but we'll talk about that later. But in the HIV virus, there's an enzyme called reverse transcriptase, and you must know this for the exam. And the enzyme reverse transcriptase allows RNA to code for DNA. And that, my friends and colleagues, is a violation of the central dogma of molecular biology. Prior to that point, we thought DNA coded for RNA, and that was it. But with reverse transcriptase, you can use RNA to code for a DNA product. Reverse transcriptase, very important for the exam, students. They're going to ask you a question about the HIV virus on the exam. I don't know what question they're going to ask you. They might ask you how to treat it, how to diagnose it, or they might ask you about the molecular biology of it. But it's always tested on the exam. Okay, so now RNA and protein. RNA and DNA are not the same thing. They have different names, they're found in different places in the cell, and they are different. So you need to understand the differences between DNA and RNA. Okay, so that's what we're going to talk about now. RNA is very small compared to DNA. Okay, you have in all the cells of your body, except for sex cells, sperm and egg cells, you have 46 chromosomes in all the cells of your body. But on each chromosome, you have thousands of genes, and each one of those genes codes for RNA. So on one chromosome, which is a huge molecule, you have 1,000 genes about, and each one of those genes codes for a piece of RNA. So what I want you to understand is pieces of RNA are very, very, very small relative to chromosomes. So RNA is very small compared to DNA, and that's going to become important when we start talking about molecular biology techniques later on in this lecture series. Okay, so reverse transcription is a characteristic of a retrovirus like the HIV virus. Okay, you need to know that for the exam. But we also have the ability in the enzyme in humans called telomerase to use RNA to code for DNA, and that's going to come up. That's going to become important in the next couple chapters. So moving right along, central dogma. Now nucleic acids, DNA and RNA differ in their length, how long they are, and we've already talked about the fact that DNA molecules are very large relative to RNA molecules. They differ in where they're located. Students, where is DNA located in a cell? Only. The only place where DNA is in a nucleus. That's right. So you're all typing nucleus. Okay, good. And you're right, Jennifer, in the mitochondria also. That's exactly right. But that's kind of an exception to the rule, and we'll talk about that later. Okay, so let me ask you another question. Where is RNA located in the cell? Nucleus and cytoplasm. Yes, Raperna, I like how you did that. N, C. Nucleus and cytoplasm. That's exactly right. So if they ask you a question about DNA, it has to be in the nucleus unless something went wrong. But RNA can be found in the nucleus and in the cytoplasm, and we're going to talk about that in the next couple days. We talked about the size of RNA and DNA molecules. DNA molecules are huge, very, very large, and RNA molecules are small relative to DNA molecules, and what they're composed of. What DNA particles are composed of is similar but not the same as what RNA particles are composed of. What is DNA composed of? ATGC, the nucleotides adenine, thymine, guanine, and cytosine. What is RNA composed of? AUGC. Now, this is a complex diagram. We're going to spend some time on this diagram. Now, DNA in your cells of your body is remember, DNA molecules are huge. And let's just review for a second now how many chromosomes are in most of the cells of your body. Not sex cells, sperm or egg cells, but the rest of the cells of your body. How many chromosomes are in? 46. That's right. You got 23 chromosomes, 2 copies of each of those, for a total of 46. You got 1 copy from mom, 1 copy from dad. That's necessarily true unless something went wrong. Now, those chromosomes are arranged from longest to shortest. So chromosome number 1 is the longest chromosome. Chromosome number 22 is the shortest. Chromosome number 23 is the sex chromosome. So chromosome number 1, if you just made it linear DNA, would be this long, 5 centimeters long, this long. So how do you get a chromosome this long into a cell that's teeny tiny? 5 trillion cells of your body. Every cell in your body has 2 copies of chromosome 1 in the nucleus. How do you get it in there? You don't have it in a linear form. You wrap it up very tightly in 3 dimensions. And that's what we're going to talk about in this diagram. So DNA, the molecule, does not exist as a linear molecule. DNA exists in a highly organized 3-dimensional structure referred to as chromatin. Now, let's talk about how that's organized at the molecular level. Well, DNA is arranged in what are called nucleosomes. So, if you look at this, this is, let's break this down here. This blue line here throughout these diagrams represents DNA, and this brown represents histones. Okay? So, this whole structure here is referred to as chromatin. So stick with me, students. Don't let me lose you. Now, if you take one of these particles here, right there, and you magnify it so you can see what it's made of, which is been done in this diagram for you right here. So this is this magnified. So look at this. You have DNA that's wrapped once, twice, around these structures here. What are these structures called? They are called histones. Okay? You have histone 3, histone 2b, histone 4, and histone 2a. And you have, in fact, 2 copies of each of those. So stick with me, students, okay? I know this is getting kind of detail-oriented. H3 here and here, H2b here and here, H2A here, and H2B here, and on the other side of this diagram you can't see it. Okay, so Vaish, you're exactly right. In this structure called a nucleosome, this is called a nucleosome right here. In that structure we have 2 copies of H2A, 2 copies of H2B, 2 copies of H3, and 2 copies of H4. And if you wrap DNA, this is DNA right here, this blue line, if you wrap DNA around that once, twice, you have a nucleosome. Does that make sense? Okay, so you have nucleosome, 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 nucleosome. All of these are nucleosomes, and it is composed of, you need to know this for the exam, DNA wrapped twice around 2 copies of H2A, H2B, H3, and H4. Now, your next question might be, why is this nucleosome so far away from this nucleosome? And this one is so far away from this nucleosome. Okay? In order to get this nucleosome, that's what I'm going to talk about right now, Maria. In order to get this nucleosome to associate closely with this nucleosome, like these up here are, you need histone H1. So here's the important thing. Histone H1 is not part of the nucleosome. That's why people have trouble understanding it. Histone H1 is not part of the nucleosome. It's outside of the nucleosome. But histone H1, when it's present, allows this nucleosome and this nucleosome to come together and interact closely with one another. Does that make sense, Maria? Yes. Close association between two different nucleosomes requires histone H1. Okay, so nucleosome, nucleosome, nucleosome without histone H1, but if you have histone H1, as is shown here, outside of the nucleosome, they can associate with each other closely. That's exactly right, Vaish. Close association needs H1. Okay, now, let's talk about this. How big are these structures? They're very small. This chromatin is about 30 nanometers in diameter. That's 30 billionths of a meter. And this nucleosome right here, from here to here, is about 10 nanometers. So 10 billionths of a meter. Does that make sense? I have seen this question on the exam. They ask you how big histone is, or sorry, how big a nucleosome is. It's 10 nanometers. I've seen that question. Now, students, I wouldn't ask that question. I don't like memorization and I don't like details, but I'm not writing the USMLE Step 1 for you. Okay? All right. Yes, Igor, but that's beyond the scope of the exam, so we won't talk about it. Yep. There's one histone, H1, for each nucleosome. Yep, they won't ask you that question. They won't ask you that question, but for each nucleosome, there's one histone H1. So there would be one here, one here, one here, one here, etc. They're not going to ask. I haven't seen that question. Now, students, when I say that's not on the exam, I'm basing that on my experience of what has been on the exam in the past and what's in your Q banks, UWorld, Kaplan Q bank. If I've never seen the question in Kaplan Q bank, then I tell you it's not on the exam. Okay. All right. So that's where I'm getting that information. So any questions on this diagram? So basically, a nucleosome is DNA wrapped once, twice around histones, 8 histones, 2 copies of H2A, 2 copies of H2B, 2 copies of H3, 2 copies of H4, and if you want 2 nucleosomes to come close together, you need histone H1. Okay? Comfortable? So this has been on the exam before, and this has been on the exam. So you need to know those sizes, how big those structures are, chromatin and a nucleosome. Okay, my friends, how are you doing? Moving right along. Now, here's a question for you. What's 30 nanometers? Ariva, this, the diameter of chromatin, right here. This is chromatin. It's condensed DNA. Yep. Okay, now what amino acids dominate histones? Why do you say positively charged, Bill? Because if you want proteins to closely associate with DNA, what's the charge on DNA? DNA is negative because of the phosphate backbone. So what charges should you have on a protein if you want it to associate with DNA? Positive charges. So you're exactly right, students. Lysine and arginine. Lysine has a positive charge. Arginine has a positive charge. You're going to see this diagram in biochemistry with Dr. Turco. Okay. What amino acids dominate histones? Lysine and arginine, because they have positive charges. Okay? All right, moving right along. Why? Because DNA has a negative charge because of the ribose phosphate backbone. So 2 copies of each histone is a histone octamer or nucleosome. 2 copies of each histone is a histone octamer or nucleosome. What does that mean? It means in this nucleosome, there's 2 copies of each type of histone. 2 copies of H2A, 2 copies of H2B, 2 copies of H3, 2 copies of H4. We've already been through that. Let me know if you have questions on that. Okay, now, actually, in 3 dimensions, it gets a little more complicated than that, but I don't think they're going to ask you it on the exam because I haven't seen that question. So stick with me, my students and my friends. This is a nucleosome right here, and then this is chromatin. This is condensed nucleosome, okay? So let's go to this one here. Next diagram. Now this is just double-stranded DNA, very small. If you take DNA and associate it in the form of nucleosomes, you get this. But what's missing here? Histone H1. But if you have histone H1, it looks like this or like this. Okay? So this diagram here is basically the same as the diagram on the previous page. If you only have nucleosomes, the diameter of the DNA is 10 nanometers. But if you add histone H1, then it's chromatin, it's 30 nanometers in diameter. But in order to do it like this, you need histone H1. Now, in reality, in the cells of your body, Igor, I've seen that question, so I would say yes. You need to memorize the histones because I've seen that question on the exam. Good question. But actually, in your body, this 30 nanometer chromatin becomes a fiber, and you can arrange that in 3 dimensions around what are called scaffolding proteins to form this structure, and you can arrange this fiber in 3 dimensions like this. But let me tell you something. The USMLE Step 1 is only going to test you from this point to this point to this point. I don't think they're going to test you on this structure or this structure. I've never seen those questions. But here's what I want you to remember, students. Just remember that it's a little bit more complex. In reality, the structure of DNA in 3 dimensions is a little bit more complex than 30 nanometer chromatin.